flower, also known as a bloom or blossom, is the reproductive structure found in flowering plants. Flowers consist of a combination of vegetative organ sepals that enclose and protect the developing flower, petals that attract pollinators, and reproductive organs that produce gametophytes, which in flowering plants produce gametes. The male gametophytes, which produce sperm, are enclosed within pollen grains produced in the anthers. The female gametophytes are contained within the ovules produced in the carpels. Most flowering plants depend on animals, such as bees, moths, and butterflies, to transfer their pollen between different flowers, and have evolved to attract these pollinators by various strategies, including brightly colored, conspicuous petals, attractive scents, and the production of nectar, a food source for pollinators. In this way, many flowering plants have co-evolved with pollinators to be mutually dependent on services they provide to one another. In the plant's case, a means of reproduction; in the pollinator's case, a source of food. When pollen from the anther of a flower is deposited on the stigma, this is called pollination. Some flowers may self-pollinate, producing seed using pollen from a different flower of the same plant, but others have mechanisms to prevent self-pollination and rely on cross-pollination. When pollen is transferred from the anther of one flower to the stigma of another flower on a different individual of the same species, self-pollination happens in flowers where the stamen and carpel mature at the same time and are positioned so that the pollen can land on the flower's stigma. This pollination does not require an investment from the plant to provide nectar and pollen as food for pollinators. Some flowers produce diasporas without fertilization. After fertilization, the ovary of the flower develops into fruit containing seeds. Flower is from the Middle English flower, which referred to both the ground grain and the reproductive structure in plants, before splitting off in the 17th century. It comes originally from the Latin name of the Italian goddess of flowers, Flora. The early word for flower in English was blossom, though it now refers to flowers only of fruit trees. The morphology of a flower, or its form and structure, can be considered in two parts: the vegetative part, consisting of non-reproductive structures such as petals, and the reproductive or sexual parts. A stereotypical flower is made up of 4 kinds of structures attached to the tip of a short stalk or axis, called a receptacle. Each of these parts or floral organs is arranged in a spiral called a whorl. The 4 main whorls are the calyx, corolla, androecium, and gynoecium. Together the calyx and corolla make up the non-reproductive part of the flower called the perianth, and in some cases may not be differentiated. If this is the case, then they are described as tepals. The androecium, or stamens, is the whorl of pollen-producing male parts. Stamens consist typically of an anther, made up of 4 pollen sacs arranged in 2 thecae, connected to a filament, or stalk. The anther contains microsporocytes which become pollen, the male gametophyte, after undergoing meiosis. Although they exhibit the widest variation among floral organs, the androecium is usually confined just to 1 whorl and to 2 whorls only in rare cases. Stamens range in number, size, shape, orientation, and in their point of connection to the flower. In general, there is only 1 type of stamen, but there are plant species where the flowers have 2 types, a normal one and one with anthers that produce sterile pollen meant to attract pollinators. The gynoecium, or the carpels, is the female part of the flower found on the innermost whorl. Each carpel consists of a stigma, which receives pollen, a style, which acts as a stalk, and an ovary, which contains the ovules. Carpels may occur in 1 to several whorls, and when fused together are often described as a pistil. Inside the ovary, the ovules are attached to the placenta by structures called funiculi. Although this arrangement is considered typical, plant species show a wide variation in floral structure. The four main parts of a flower are generally defined by their positions on the receptacle and not by their function. Many flowers lack some parts, or parts may be modified into other functions or look like what is typically another part. In some families, such as the grasses, the petals are greatly reduced. In many species, the sepals are colorful and petal-like. Other flowers have modified stamens that are petal-like. The double flowers of peonies and roses are mostly petaloid stamens. Many flowers have symmetry. When the perianth is bisected through the central axis from any point and symmetrical halves are produced, the flower is said to be actinomorphic or regular. This is an example of radial symmetry. When flowers are bisected and produce only one line that produces symmetrical halves, the flower is said to be irregular or zygomorphic. If, in rare cases, they have no symmetry at all, they are called asymmetric. In those species that have more than one flower on an axis, the collective cluster of flowers is called an inflorescence. Some inflorescences are composed of many small flowers arranged in a formation that resembles a single flower. A common example of this is most members of the very large composite group. A single daisy or sunflower, for example, is not a flower but a flower head, an inflorescence composed of numerous flowers. An inflorescence may include specialized stems and modified leaves known as bracts. The transition to flowering is one of the major phase changes that a plant makes during its life cycle. The transition must take place at a time that is favorable for fertilization and the formation of seeds, hence ensuring maximal reproductive success. To meet these needs, a plant is able to interpret important endogenous and environmental cues such as changes in levels of plant hormones and seasonable temperature and photoperiod changes. Many perennial and most biennial plants require verbalization to flower. The molecular interpretation of these signals is through the transmission of a complex signal known as florigen, which involves a variety of genes, including constants, flowering locus C. In flowering locus T, florigen is produced in the leaves in reproductively favorable conditions and acts in buds and growing tips to induce a number of different physiological and morphological changes. The first step of the transition is the transformation of the vegetative stem primordia into floral primordia. This occurs as biochemical changes take place to change cellular differentiation of leaf, bud, and stem tissues into tissue that will grow into the reproductive organs. Growth of the central part of the stem tip stops or flattens out, and the sides develop protuberances in a whorled or spiral fashion around the outside of the stem end. These protuberances develop into the sepals, petals, stamens, and carpels. Once this process begins, in most plants, it cannot be reversed and the stems develop flowers, even if the initial start of the flower formation event was dependent of some environmental cue. The principal purpose of a flower is the reproduction of the individual and the species. All flowering plants are heterosporous, that is, every individual plant produces two types of spores. Microspores are produced by meiosis inside anthers, and megaspores are produced inside ovules that are within an ovary. Anthers typically consist of 4 microsporangia, and an ovule is an integument megasporangium. Both types of spores develop into gametophytes inside sporangia. As with all heterosporous plants, the gametophytes also develop inside the spores. Since the flowers are the reproductive organs of the plant, they mediate the joining of the sperm, contained within pollen, to the ovules contained in the ovary. Pollination is the movement of pollen from the anthers to the stigma. Normally, pollen is moved from one plant to another, known as cross-pollination, but many plants are able to self-pollinate. Cross-pollination is preferred because it allows for genetic variation, which contributes to the survival of the species. Many flowers depend on external factors for pollination, such as the wind, water, animals, and especially insects. Larger animals such as birds, bats, and even some pygmy possums, however, can also be employed. To accomplish this, flowers have specific designs which encourage the transfer of pollen from one plant to another of the same species. The period of time during which this process can take place is called anthesis, hence the study of pollination biology is called anthecology. Flowers that use biotic vectors attract and use insects, bats, birds, or other animals to transfer pollen from one flower to the next. Often they are specialized in shape and have an arrangement of the stamens that ensures that pollen grains are transferred to the bodies of the pollinator when it lands in search of its attractant. In pursuing this attractant from many flowers of the same species, the pollinator transfers pollen to the stigmas, arranged with equally pointed precision of all of the flowers it visits. Many flowers rely on simple proximity between flower parts to ensure pollination, while others have elaborate designs to ensure pollination and prevent self-pollination. Flowers use animals including insects, birds, bats, lizards, and even snails and slugs. Plants cannot move from one location to another, thus many flowers have evolved to attract animals to transfer pollen between individuals in dispersed populations. Most commonly, flowers are insect-pollinated, known as entomophilous, literally insect-loving in Greek. To attract these insects, flowers commonly have glands called nectaries on various parts that attract animals looking for nutritious nectar. Some flowers have glands called eleophores, which produce oils rather than nectar. Birds and bees have color vision, enabling them to seek out colorful flowers. Some flowers have patterns, called nectar guides, that show pollinators where to look for nectar. They may be visible only under ultraviolet light, which is visible to bees and some other insects. Many flowers have close relationships with one or a few specific pollinating organisms. Many flowers, for example, attract only one specific species of insect, and therefore rely on that insect for successful reproduction. This close relationship an example of convolution, as the flower and pollinator have developed together over a long period of time to match each other's needs. This close relationship compounds the negative effects of extinction. However, since the extinction of either member in such a relationship would almost certainly mean the extinction of the other member as well. Flowers that use abiotic, or non-living, vectors use the wind or, much less commonly, water, to move pollen from one flower to the next. In wind-dispersed species, the tiny pollen grains are carried, sometimes many thousands of kilometers, by the wind to other flowers. Common examples include the grasses, birch trees, along with many other species in the order Figales, ragweeds, and many sedges. They have no need to attract pollinators and therefore tend not to grow large, showy, or colorful flowers, and do not have nectaries, nor a noticeable scent. Because of this, plants typically have many thousands of tiny flowers which have comparatively large, feathery stigmas to increase the chance of pollen being received. Whereas the pollen of entomophilous flowers is usually large, sticky, and rich in protein, anemophilous flower pollen is typically small-grained, very light, smooth, and of little nutritional value to insects. In order for the wind to effectively pick up and transport the pollen, the flowers typically have anthers loosely attached to the end of long thin filaments, or pollen forms around a catkin which moves in the wind. Rarer forms of this involve individual flowers being movable by the wind, or even less commonly the anthers exploding to release the pollen into the wind. Flowers can be pollinated by two mechanisms: cross-pollination and self-pollination. No mechanism is indisputably better than the other, as they each have their advantages and disadvantages. Plants use one or both of these mechanisms depending on their habitat and ecological niche. Cross-pollination is the pollination of the carpel by pollen from a different plant of the same species. Because the genetic makeup of the sperm contained within the pollen from the other plant is different, their combination will result in a new, genetically distinct plant through the process of sexual reproduction. Since each new plant is genetically distinct, the different plants show variation in their physiological and structural adaptations, and so the population as a whole is better prepared for an adverse occurrence in the environment. Cross-pollination, therefore, increases the survival of the species and is usually preferred by flowers for this reason. Self-pollination is the pollination of the carpel of a flower by pollen from either the same flower or another flower on the same plant, leading to the creation of a genetic clone through asexual reproduction. This increases the reliability of producing seeds, the rate at which they can be produced, and lowers the amount of energy needed. But, most importantly, it limits genetic variation. In addition, self-pollination causes inbreeding depression, due largely to the expression of recessive deleterious mutations. Following the formation of zygote, it begins to grow through nuclear and cellular divisions, called mitosis, eventually becoming a small group of cells. One section of it becomes the embryo, while the other becomes the suspensor, a structure which forces the embryo into the endosperm and is later undetectable. Two small primordia also form at this time, that later become the cotyledon, which is used as an energy store. Plants which grow out one of these primordia are called monocotyledons, while those that grow out two are dicotyledons. The next stage is called the torpedo stage and involves the growth of several key structures, including the radicle, the epicotyl, and the hypocotyl. In the final step, vascular tissue develops around the seed. Many flowering plants reflect as much light as possible within the range of visible wavelengths of the pollinator the plant intends to attract. Flowers that reflect the full range of visible light are generally perceived as white by a human observer. An important feature of white flowers is that they reflect equally across the visible spectrum. While many flowering plants use white to attract pollinators, the use of color is also widespread. Color allows a flowering plant to be more specific about the pollinator it seeks to attract. The color model used by human color reproduction technology relies on the modulation of pigments that divide the spectrum into broad areas of absorption. Flowering plants, by contrast, are able to shift the transition point wavelength between absorption and reflection. If it is assumed that the visual systems of most pollinators view the visible spectrum as circular, then it may be said that flowering plants produce color by absorbing the light in one region of the spectrum and reflecting the light in the other region. With CMYK, color is produced as a function of the amplitude of the broad regions of absorption. Flowering plants, by contrast, produce color by modifying the frequency of the light reflected. Most flowers absorb light in the blue to yellow region of the spectrum and reflect light from the green to red region of the spectrum. For many species of flowering plant, it is the transition point that characterizes the color that they produce. Color may be modulated by shifting the transition point between absorption and reflection, and in this way a flowering plant may specify which pollinator it seeks to attract. Some flowering plants also have a limited ability to modulate areas of absorption. This is typically not as precise as control over wavelength. Humans observers will perceive this as degrees of saturation.